There's our field of view when we're looking at the endospore stain with the 40x total magnification. And when we go up, there you have it with 100 times total magnification. And I'm just going to go over and remember, since we're looking at our bacteria cells, we're going to add our drop of immersion oil by straddling the stage with your objectives there, and just put your drop of immersion oil right over the center one and swing that objective so that it is touching the oil. And here we can see the results from one of our endospore stains, and you can see that nice sort of bluish green or cyan color. But the one thing that is lacking from scanning around here is the cell bodies, and here we can see a few of them. And when you see a field where the majority of the specimens are lacking those cell bodies, what's gone on here is more than likely all of the cells that were in there actually went ahead and did this process of sporulation. So we can't really speculate about the cells from this particular specimen unless we can find a red cell body with this spore on the inside. And so I'm going to scan around and see if I can find. So I found a field with a few of them in here, and I'm sort of focusing past them to try and pull up some of that pink. But what we want to do is try and gauge where these endospores are relative to the cell body. And so we can definitely tell the shape of them. And over here, it looks like we can see a few of them inside the body of the cell there. And so that's what we want to look for, is to try and gauge the size of our cells and also the shape and the location of the endospores that are inside. The majority of what we see here, though, are exospores. But right there, we can sort of see some of the cell bodies right below the 50 there. So there are some endospores in there, but the majority of this specimen are exospores. On this specimen, it looks like the opposite problem is occurring. These guys look like they did not make a lot of spores from scanning around. There's really only a few of them that appear to have the green. The majority of what we're seeing here is just the cell bodies that have picked up the counter stain from the end there with the saffron in. So we could measure these against the ruler and get some idea about the size of them, and you can sort of see there is a little bit of green in some of these, but not as distinct as I would like. I would like to see something that's sort of in between with the last one there. So maybe our unknown from the soil will allow us to see those. So here is our soil unknown, and you can see from this clump of cells that we do have some cells and we do have some spores. And then it's a matter of finding a good field where you can actually measure both the size of the cell and also determine the location of the endospore within the body of the cell here. And so if we look at this one in the center here, sort of above the 50, there's a nice one. Looks like we can actually see some of the body of the cell, and you can also see the spore inside of the body of the cell there. And like I said, you can also see a lot of cells scattered throughout the field that don't have endospores, and that's fine. Those would be your living cells. Not all cells are going to make spores, even if they have the potential to do it. They need some sort of triggers sometimes. So there we can see some examples of our endospore stain.